I would have, in the case of a floppy disk, 1.4 megabytes of information stored in the disk. Again, all that happened is that the constituents on the floppy disk were reordered in order to provide and store information. Knowing this then, that information is a non-material entity, then how do we explain the information we find in DNA? Now what we have in DNA which makes information is sequence specificity. So the sequencing of constituents provides information, and in this case it's the sequencing of nucleotides. The order of nucleotides in a DNA chain is what provides the blueprints which carry information which order and direct the ordering of life and the creation of life. Now, in order to try to explain this naturalistically, naturalist presuppositional scientists have argued what's called a self-organizational scenario, or some scenario where law-constrained chance was sufficient to order and constrain the nucleotides in order to cause them to be in the right order to provide information. But this type of theory fell critically apart upon the discovery that there is no critical chemical bonding between nucleotides in their sequencing order. The chemical bonding is solely, or the critical bonding is solely, between the nucleotide and the spine of the DNA. The illustration given of this is the magnetic letters that children play with on refrigerators. The chemical attraction, or the natural attraction, is between the magnet and the refrigerator. So if a child were to write on a refrigerator with his letters C-A-T, cat, or I-N-F-O-R-M-A-T-I-O-N, information, we would know that the chemical properties, or the natural law properties, are between the magnet, the letter, and the refrigerator, not in the order in which the letters are placed in the fridge. In fact, nothing can influence the order except the child, the intelligence of the child writing and placing the information on the fridge. Dr. Francis Crick likened these to typeset, letters on a typeset. Just as in old typeset printing, the typeset would have a block, and it'd have a letter imprinted on the block, and then the printer would take those typesets, place them on his printer, and press them down on a page. Amino acids work the same way. The spine of the amino acid is the only place where chemical bonding occurs. So that along these spines of the amino acid, which are all chemically identical, we have a corresponding binding. But the information isn't in the binding part of the amino acid. It's in the side chain of the amino acid. Whereas the side chain would be considered the same as the imprinted letter onto the typeset block. So that if you put the typeset block properly into the typesetting machine, it's simply a square block. It doesn't govern what letter goes where. In the same way, in an amino acid chain, what governs where the amino acids bind is simply the spine. But no information is carried in the spine. The information is carried in the side chains. And then the sequence specificity of the amino acid side chains, the order in which they go, is where the information of life is found. And there is no governing chemical attraction which dictates which amino acid goes in what sequence. That information is not found in any kind of critical chemical bonding. It's not required by any kind of critical chemical bonding, and yet it exists as specified complexity, which again is a clear pointer, an empirical marker to actual design. So then again, what we're arguing here is a system of free inquiry, a competing hypotheses, best explanatory model system. So here are our four models of explanation. First, we have the explanation of necessity. Second, we have the explanation of chance. Third, we have the explanation of chance and necessity, or law-modified chance. And fourth, we have the explanation of intelligent design. We've seen that chance is insufficient to explain the complexity in DNA. We find that law is insufficient because there's no critical chemical bonding between the nucleotides, and natural chemistry can't separate amino acid isomers. we find that constrained chance, or chance modified by law, also fails. Remember that in evolutionary theory, natural selection is the constraining law. However, natural selection cannot function without a complex reproducing system. At this point in biogenesis, a reproducing system doesn't exist. Natural selection is therefore a non-starter and an unavailable answer. There is no natural law that can constrain the informational sequencing of nucleotides in biogenesis. Therefore, what we have here, again, design theory, comes across as the best hypothesis with the most explanatory power, taking all of the evidence. Specified complexity, a clear marker of intelligent agency. So we'll take an examination now also of chaos theory, because chaos theory is the alternate self-organizational scenario which is offered. In order to better understand chaos theory, take a look at the placement of pitches from this pitching machine. From the batter's perspective, there would seem to be an unpredictable randomness to the location of the pitches. But are the pitches actually random, or could they be predicted given perfect knowledge of the initial conditions? For example, if we were to know the texture of the ball, the exact speed of the wheel spinning on the machine, the exact voltage and amperage to the machine, the density of the air through which the ball must pass, any breezes or winds, the amount of contact between the ball and the wheel, the amount of friction between the ball and the wheel, the temperature of the ball and the machine, and so on through many, many other initial conditions which govern the flight of the ball. If these were perfectly knowable and predictable, then theoretically, the outcome could be absolutely predictable and would not be random. So the randomness is apparent based upon the limits of our knowledge. And accordingly, the closer we get to the pitching machine, the greater the ability to predict the location of the pitch, because the causal variations are